introductory thing.
Yeah, we could.
Well, not yet. Oh, I'm sorry.
All right, yeah. Hey everybody, come on in, tune in, and I hope people that are joining us on YouTube, because we're starting with our announcements just a little bit earlier than normal, by 5 minutes, just because there's a lot of exciting information to get through. This is the February meeting of the Madison Astronomical Society, and it's great to see everyone here. At around 7:30, we're going to have our guest presentation tonight that I'll tell you about a little later. And also, we've got our usual type of announcement thingies, but this month we've got a little bit of extra exciting information. A project that we've been working on a couple of years now, as far as increasing the number of concrete pads and electrical outlets at our dark sky observing site, Yana Research Station. Ever since it's been about 10 years since I first joined the club, and our membership, the number of people with memberships has essentially doubled since the time I've been a member of the club. And before the pandemic, there were some times where we'd go out on a clear night in the middle of summer at Yana Research Station, and there would be more people with their telescopes there than we had electrical outlets and concrete pads. So with the growth of the club, the board of directors realized that some of the outlets don't work so well. Sometimes people are using some of the outlets and they go out unexpectedly, and the electrical outlet and the privy, the electrical tripper for that, the circuit breaker for that goes off from time to time. So we're like, man, we'd really like to modernize all this infrastructure here and kind of propel our Yana Research Station into the next few decades as far as being an active and dark sky observing site that members in the area can feel welcome to use and have plenty of resources to use that dark sky out there. So I would like to introduce our club treasurer, Jürgen, who has been worked very diligently on making some very detailed plan, and I'll just hand it off to you, Jürgen, to kind of explain what we're working on.
Okay, I'm Jürgen Patel, I'm the treasurer. I take your money happily. I started out, this is what YRS is. And those of you who've been out there probably have not really seen this view unless you go to Google Maps. This is the clubhouse. People come in from here. This is Kelly Road. And we have right now 6 observing pads and 2 places where you can get power. And these 2 where you get power come from 1 circuit breaker. Okay? So that's a problem. I went out there one night to try to do some observing, and I couldn't do anything because my mouth took too much power. This is the DGRO, Dr. Greiner Research Observatory. This is the .G, and this is the AKO, and buried in the weeds is the privy. This is the deck, the handicap deck that was built some years back. And this used to be a roll-off where there was a big, I think it was a big daub in there, I'm not sure exactly what was in there, that we took out. And right now it's just simply a concrete pad. There are 3 other power outlets here, here, and 1 back here, which is absolutely useless. And that's what we're faced with. As Lawrence said, we have too many people for the resources we have, and we have too poor power for people to use because more and more is observing where you need power, or I'm sorry, astrophotography, where you need power, or even if you're doing visual observing, you need power to guide your scope. Anyway, so the MAS board actually realized we had unmet needs back in 2019, and we started thinking about all of this. We created the YRS Upgrade Committee, which is made up of 10 people, actually 11, which is a board of directors plus 3 heavy-duty observers out there. You may know them, Avtar Rupra, Bob Hamers, and Jeff Schockler. So we ran into a number of concerns that I won't repeat them because Lawrence already said them. And if my mouse works? Yeah, it does. So this is actually what we did. The issues we had were more people, more requirements for electric power, and the rise of large daubs. Where does that come in? Daubs are long tubes. And if you want to put those on a 6 by 6 foot pad, you run out of room. So the obstacles are too few pads, no larger pads, limited outlets, and there's small circuit breaker panel. This is a two-phase approach to upgrading YRS. The first phase was to replace, the first phase was to replace the small circuit breaker with a 200 amp panel and add space for more circuit breakers for phase 2. And then we also have uncharted wiring. Now, everything that's out there right now, the pads and the wiring, were done by volunteers. And that was great. Saves a lot of money. But it also means we don't have a lot of documentation on what's out there. We may have had it one time, but you know what happens with records. So this is the existing layout. This is a diagram you'll see in several forms coming up. It's the clubhouse, the 6 pads, the 2 electrical outlets powering the pads, electrical outlets here, I missed that one, and one over here, the clubhouse, of course, and the privies down here. The line around that is what essentially is a squared off boundary of the tree and shrub line. And that's the space in which we can play. That's really what I call the observing area. Yeah. So the issues that the committee had to come up with was location of additional pads, the number of pads we're going to put in, how to deliver power to pads, grounds restoration, because this is going to make a mess out at YRS, and then also the vendor selection. Now, we picked Hill Electric to do the electric panel upgrade, so we stayed with them. We talked to them about contractors that they have had good experience with, good relations with, and we picked Joe Daniels. It's not necessarily the cheapest approach, but they're reliable. Notice I didn't say anything about cost yet. The new pads, what we plan to do, or what the committee's plan came up with, is this is the old drawing, and I've overlaid it with green for the, there we go. These are the new pads, there are 14 new pads. There's going to be power to each pad. We have a 200 amp panel. If all 14 are drawing 15 amps per, in trouble, but I rather doubt that. And our electric meter will go round and round. We'll probably have to increase rates just to account for that, but that'd be a nice problem to have. The electrical wiring, every bit of wiring out at YRS, underground wire, is being abandoned. We're going to have Hill come in, they're going to trench, they're going to put wiring in conduit, they're going to document what they do, so hopefully we'll have good plans afterwards. Another thing, if you notice, each of these is larger than the one it's replacing. We are going from 6 by 6 foot pads to 7 by 7. By doing that diagonally, you actually add about another 1.5 feet. So for long scopes, that should be a good thing. We have picked, and we did this really just because we had to put it in. We picked the northwest, by the way, that is north. The northwest corner of each pad is where the electricity is going to be. It's going to be, you'll see a diagram coming up, but it's going to stand about this high. If that's 16 inches, it's going to stand about this high, and it will occupy roughly 1 square foot in the corner. Oh yeah, and the spacing between the pads north and south is 6 feet. That's what it had been. None of the interpad spacing is changing. The distance between pads east-west is 10 feet. That's what we've had all along, and it seems to have worked out very well. Also, the pads are going to be level. They're going to be at ground height, so there won't be any stumbling. Of course, I will always find a way to stumble, but we won't go there. But two things are disappearing. This roll-off pad, which right now is just a broken concrete pad, is going away. That'll be taken away. And the deck is going away. Now, the deck has been there for quite a while. A lot of time and effort has been spent maintaining it, but it always needs effort and maintenance. There's no scope in here. There's actually a Rubbermaid hut here, or little cabinet, and it's got a pier in it. No scope is mounted on it. Every time we have a cleanup day out there, we go in and clean out bugs. Other than that, it hasn't been used. I've been treasurer for 6 years. It hasn't been used in the time I've been treasurer.
Yeah, bugs, snakes, and mice usually.
Yeah, and all the stuff that they leave behind. So it's with some reluctance that we're going to take it out, but it allows us a couple more pads. And the new breaker panel, the reason we put in a big one is it feeds all the new stuff. Everybody is getting it. We're going to add a second wire coming into the DGRO. We're going to actually put in its own line to the privy. So if somebody can figure out how to use 15 amps in the privy, more power to them.
Need a toilet seat.
Toilet seat, yes, thank you.
Bidet.
I originally toyed with the idea of putting in 17 pads, but we are leaving the Dachi there. We have good reason to keep it. And so 2 of these pads, and this one is simply too close to the AKO, so we're not doing anything with it. But these are the pad structures that we have. If you take a look at this, the pads here and here and here have pretty much unobstructed west views. Right now, some of these pads are actually limited in how high off the horizon you can get by the roof. However, these pads here aren't going to see, they're only about 10 feet. So you're not going to see a lot. However, you get great views. So everybody's going to have an option. You know, what do you want to look at? The one thing you can't look at is the east. There's a tree line here.
Yeah, the rows closest to the clubhouse are going to be your best bet for looking east, pretty much.
Yeah. This is the same diagram, just with a little bit of notation. The driveway is down here, the tree line is down here. We had the tree line down here top this year, so you actually have better. And the distance here is the same as 21 feet 3 inches, if my measurements are right. We're going from 21 feet here to only 10 feet. That's why you'll actually see the roof of the DGRO. Here's what the unistrut mounts look like.
Yeah, these for the electrical outlets.
These are for the electrical outlets.
For each pad.
Yep. Our model is Aaron Ramberg. He's the rep from Hill Electric. We looked at putting in, as we have right now, wooden piers, wooden 4x4s. But the trouble is those add several $100s per. And so 14 of those would add about another $3,000. So we're not going to go for those. Also, any other pier would be outside the concrete pad. You put them outside the concrete pad and you run into problems because now you've got 14 instead of 2, and the poor guy who mows our lawn is going to go crazy. These are set back about 8 inches from the edge because you don't want to hit them and break the concrete. The concrete is going to be nice and strong, but you want to have these far enough back that they won't damage. What's missing here is the plastic cover, so that sticks out. So the total distance, the diagonal distance going in is about 1 foot to 13 inches. And that's the amount of space this will take.
Jürgen, I'd also like to add that one of the downsides of having the outlets in the grass between is it creates a trip hazard.
Yes.
If you have wires in the grass between that post and the pads, people are tripping all over the place. This way, all the wires are right there on the concrete, and if you're on the concrete, you know it because it's your pad.
Yeah, and I appreciate that because I meant to say that. Thanks for catching it. Making sure we don't have any tripping hazards is a big deal here because this is all at night. We don't have any lights, hopefully.
Yeah, and before this thing goes final and we actually build these things, we do plan to do a quick mock-up just to make sure that it's comfortable for someone who's got a tripod with a large telescope and doing visual observing, or someone with a large job, that that would be a space that people would be comfortable in and not worrying about running into that unit strut. But.
One thing about this is that the biggest source of controversy about the upgrade project has been the design of this.
Yeah, and I want to add that even with this in position taking up a part of the pad, the remaining space on the pad is as big as the existing pads are. So if your equipment fits on the 6x6 pads now, it will fit on the 7x7 with the electrical.
And if you're worried about something hitting that, move it a foot back, because that's all it takes, and it'll clear it. The only time you'll probably run into problems is when you're closing up for the night. You've got your huge daub lying level, but hopefully if we put this in the same position on each pad, you'll get used to it and you'll automatically compensate for it. Dan Heslop created 3 images of what it's going to look like. Now, the one problem is, I found this fascinating. If he put the top of this down on the ground, the grass grew through the pad. So we figured just leave it like this. But this will be flush with the ground. The current drainage slope will be maintained, but this is what it would look like at ground level.
And that survey marker will still be where it is.
Bingo, that's actually an important thing.
That north pointing survey marker that has the latitude and longitude of the observing site, that will be left intact.
Yes, in fact, that was part of the proposal. They had to make sure that that thing goes back to where it was. We had 4 proposals on how they're going to dig all this stuff up, and they're going to dig these out one by one. Doesn't make any sense. Anyway.
It's probably a lot of the labor we're paying.
Yes.
Guys out there with shovels for each of these.
This is looking from the east, looking west in probably about 30 or 40 feet in the air. This is what the layout would look like. And this is what it would look like diagonally looking from southwest to northeast. So this gives you an idea of what it would look like. The only difference is it would be nice if we had those trees. But no. I'm saying this is what it looks like 100 years from now when we've got global warming. So, okay, what comes next? We are going to be, the total cost of this is about $36,700. Yeah, it's a lot of money. I had some notes on this. I had pause at this point for shock to hit. We actually could afford that right now, but it would leave us very, very, did I say very short on funds.
We would be worried about cash flow at that point.
Yes. So what we're going to do is we're going to recommend start a big fundraising campaign. And we're toying with the idea of asking for contributions of $500, and in return for that, you'll get a memorial plaque buried in the pad, in a pad. And we haven't designed that yet. We've talked to Joe Daniel. They're aware of doing this. Is not a big deal. It would be probably a ceramic pad that's embedded in the concrete with your name and a message. That design remains to be worked out. If we can sell all the pads or even most of the pads, we'll be financially okay going forward. You know, we could live, but it will be very, very tight. So and we also may have some unanticipated. I mean, that's $36,700 roughly dollars. The difference is it doesn't pay for watering the grass. There's no water out there. So we're going to have to figure out some way to water the grass after they restore it. So, does anybody have any questions at this point?
Is there a well?
No. There is a well that is owned by the previous property owner, but it runs for a short time and it goes dry. So it's kind of teacup by teacup is probably what it would be.
We don't need to dwell on the water.
No.
The board and the YRS committee have talked about the water situation. We think we'll be able to improvise something when it comes up. Big thing is we want feedback from the members.
Yes.
Tentatively, we'd like to have a general membership vote about approving this expenditure next month at the March regular membership meeting. And so we want feedback before then. And we've already had some offers on pad donations. So if you want your name on a pad, get in touch with the board soon, because who knows, they may sell out.
Yeah, our price that we've kind of agreed on with the board and based on feedback from some people who are interested in having a plaque with their name on it, we're targeting $500 donations to get a plaque. But, you know, lesser
If you want to donate more,
Yeah.
we can put a plaque in
Yeah.
the clubhouse, we can name the latrine after you, we can do anything you want.
yeah, a tattoo. Oh, yeah, a tattoo.
But
John
we,
Rummel will tattoo your name on his back.
so, any other comments or questions? No.
No. Oh, yeah, he's. I think he's watching. Well,
Okay.
if there aren't any other questions, I see we are getting towards 7:30. So,
Lawrence, I do have a question.
Oh, yes, Terry?
On that voting, are people going to have to be present?
No.
Or can they vote online also?
We've done online voting.
We've done online voting via Zoom,
Yeah.
but have we done it via YouTube?
No.
no, we can't do it via YouTube.
Well, they can,
Oh,
they can
But
state.
the problem with that is there's no way to verify who they are. The advantage when we did it via Zoom, you could see by camera that the person that was present was a member of MAS and for legal
Their
purposes.
names come up.
Right, but that does not prove their identity like the video.
Zoom, though.
Well, Zoom, if you, no, if you can see them on video,
Not everybody was on video.
but that's the, see,
If you
that's
go in
the
by
problem.
telephone, for
Yeah,
example.
that's the problem that these sorts of things can bring up, but.
We're gonna discuss that, and
Right.
we'll find some sort of alternative.
But I would highly encourage, because that's the thing, is with a month's notice, I would highly encourage for all interested parties to come here and get a quorum here, because I think the last time we had an election here, we did have a quorum here. The first year where maybe my memory's wrong, because, yeah, we only started coming back to doing meetings here just this last May, and we did have some people join us by Zoom. Yes, question?
I would recommend that you look at the bylaws to see what the provisions are for voting. They probably say you have to be in present.
Oh, there is a quorum requirement for a certain percentage of the membership,
15%.
plus and a minimum number of board members, that's 15%, you're gonna say. And a minimum number of board members must also be present.
But to address the specific question of in person, we went through this process with the bylaws when we went to virtual meetings at the beginning of the pandemic, and the board decided that by our interpretations of the bylaws, we could do it via Zoom at the time.
The thing is, it's not precluded by the bylaws,
Right.
Exactly.
right? It's not, in fact, it's not mentioned. Virtual meetings are not mentioned.
Right, and it just says the bylaws just define what a quorum is, including the minimum number of regular members and board members that must be present. And our bylaws also, in case anyone's curious, also define that there is an endowment, that we have $6,000 that we have to carry over in the account.
And by the way,
and that's money that's hands off, that's like a last resort emergency endowment money that we don't touch as a board.
Yeah.
Just want to reiterate that, like Jürgen was saying, what Lawrence just said, that we do have this endowment that we're not touching. Even if not a single person bought a pad or donated, we could afford this, it'd just be tight, and we wouldn't be touching that nest egg.
We would end up with, if nothing else changed other than the budget I've outlined, we would end up on September 30th with $2,000 to spend. And then we get a couple $1,000 for membership renewals in October, and we'd have to live on that. So that's getting awfully tight.
Yeah, and we don't want to do that.
Yeah, we don't want to.
But like we said, we already have several pads that have been, I don't want to say spoken for, but we've got quite interest in pads so far. So if you're interested in a pad or another additional donation, let us know. Because even if we only sell half the pads, that'll be enough padding to get us comfortably through the next couple of years.
And you know, if you really want to get on a pad and we're out of pads, maybe we can just have a bidding war for some of the pads. I'm joking.
I'm joking, as a joke. All right, please bear with us while we get ready for our next presentation. I'm going to switch you off quick. Just bear with us while we get ready for our guest presentation. And here's your microphone, and you can work on it. His mic, because they were saying it was crackling when he was talking. Okay, and that can happen if you put it on like that. Hopefully the microphone does not rub against the shirt. So, yep, and I'll switch you on. Sorry. There you go. Test check. Yeah, sounds like we can hear you. All right. All right, now I'm going to have to, yes, what's going on? Because
For the next 7 members, you have to contribute $500. That's the admission fee.
well, anyways, I got to get serious here because we're a little bit short on time, and yeah, we could ramble on a while. But I just would like to. We have a very special guest presentation tonight on a very fascinating topic. We have Vatsan Raman from the UW Biochemistry Department, and he's doing a presentation on Bacteriophages or phages. It's entitled, How Do Bacteriophages Interact with Their Host Bacteria in Space? And just to tell you a little bit about him, Vatsan is an associate professor in the Department of Biochemistry at the University of Wisconsin-Madison. He is an affiliate of the Department of Bacteriology and Chemical and Biological Engineering. His research group aims to develop technologies at the intersection of biochemistry, microbiology, computation, and engineering to understand the fundamental principles of biomolecular and cellular systems. Before joining the UW-Madison, he was a Weiss Technology Development Fellow at the Harvard Medical School, where he developed foundational technologies to engineer microbes using biosensors to sustainably produce fuels and chemicals. In his graduate work, he developed computational methods in the Rosetta Biomolecular Modeling software suite for predicting protein structures at the atomic level accuracy at the University of Washington in Seattle, which, if you've ever been there, is a gorgeous campus just like our own. So please, everyone here, join me in welcoming Dr. Vatsan Raman. Thank you for joining us.
Thank you.
Quick question: how would you like to have people ask you questions during or at the end of your presentation?
Oh, absolutely. No, please interrupt me. This is a nice, cozy setting. So the more questions we ask, the more fun it'll be. I don't know how much biology you generally get out here, but I have a 40-minute presentation. We can stop it halfway depending on how the interaction goes. And so I'll try to get some data towards the end, but we'll try to go easy on the data just so that there's a little more discussion. So again, I'll start off with some basics. Just to give a little more introduction, I came to the United States about 22 years from India. I came here to Missouri before I was in Seattle to get my master's degree in chemical engineering. And then I got my PhD in Seattle, and then I was at Harvard for a couple of years. And I've been in Madison now for about 7 years now. So that's me. Okay, so what I thought I'd do to get started is first I want to acknowledge the people in my lab. So these are the people who do all the cool stuff. So I'm just a spokesperson here. And so that's the benefit of running a research lab, is you get to take credit for all the stuff that people in your lab do. And so these are the people that make it all happen. Okay, I thought I'll start off with the trivia question, which is, what is the most abundant life form on Earth? You can put your hands up. Yeah. What was that? That's close, but it's not quite correct. Yeah, viruses are the most abundant life form on Earth. Yeah. Yeah, so that's the, you saw my slides. Now, there are two kinds of viruses. There are the viruses that infect humans. COVID, flu is one kind. And there are viruses that infect bacteria, which is what these bacteriophages are. And that's what I'm going to talk about for the next 20 minutes or so. And just to put a number on this, why is it the most abundant life form? The estimates are there are 10 to the 31 bacteriophages on Earth. 10 to the 31, so just sort of think about that number for a second, right? It's an exceptionally large number. Now, why is the virus the most simplest life form? That's a follow-up question. Why is it the most simplest life form? Well, close. But so the reason it's simple is because it only has its DNA, which is its molecular blueprint. It needs a host to propagate, right? So it has all the rule book on how to make more viruses, but it cannot make more viruses by itself. It has to get into a host cell, use and hack the host machinery to make many, many copies of itself. And that's how. So that's what makes it simple, because it has the rules, but it doesn't have the other chemicals and the enzymes and proteins to make more of itself. Okay. Now, so this is what a bacteriophage looks like. So I said there are two kinds of viruses, the ones that infect us and the ones that infect bacteria. And the ones that infect bacteria don't infect us. And so this is a picture of a bacteriophage. Kind of looks like a lunar lander, so all space enthusiasts here. So this looks like a lunar lander. And this is a bacterial surface. And so you can sort of imagine this as the longest evolutionary experiment that's been happening on Earth, right? So what happens is the bacteriophage, and I'll sort of interchangeably use the word bacteriophage and phage. The bacteriophage tries to get into its bacterial host to replicate or multiply, and the bacterial host.
Is there any possibility you can use your mouse pointer instead of your laser pointer, because people online can't see it?
Where is the mouse pointer? Here we go. Okay, when I get to the data part, I think I'll point with that. So you can imagine this is the longest evolutionary experiment, because the bacteriophage tries to get into the bacteria. And when it does that, it actually kills the bacteria, right? And so what the bacteria then does is it tries to sort of counteract that effect by mutating itself to prevent the bacteriophage from entering. So this constant arms race has been going on for billions of years. Now, this is an image of what it looks like. Again, this is your typical E. coli bacterium that we might see as pathogens. And so these are the tiny bacteriophages that are sitting on top of the pathogen, right? And I'll see if I can pull up a movie here. I don't know if YouTube would work. So this is an animation of this. So you can see what this bacteriophage does. It kind of launches itself and settles on the surface of the bacterium. And once it does, it injects its DNA, which is present in here, into the host bacterium. So that's what it does. And once the DNA goes inside, the bacterial host, which is this thing down here, thinks it's its own DNA and starts making all the proteins and the nucleic acids that are essentially the phage products. And when that happens, all the bacteriophages assemble inside the host. So this is the phage DNA that's now inside the host. And then it sort of tricks the host machinery to use it and make more phage progeny inside the bacteria. And then eventually what happens is you make so many of them, it kind of pops the bacterial cell open and then goes off and infects other things. So this is sort of what the life cycle of the bacteriophage looks like. Yep.
What happens to the bacteriophage that does the injection?
That's the end of its own life cycle. So it kind of sacrifices itself and makes 100 babies every time it goes through its bacterial host.
We do have requests online just to repeat the question anytime anyone
Got
asks.
it. So the question was, what happens to the bacteriophage that delivers its DNA? That's the end of its life cycle. It makes progeny bacteriophages. I'll get to the space part in a few slides, but I thought I want to sort of set the platform before I get to the space part. Okay, so this is again, sort of a schematic of what it does. Here is the bacteriophage, here is the bacteria. It sort of pushes its DNA inside, it replicates or multiplies, and then eventually sort of breaks the bacteria open, right? So this is what the life cycle looks like. Now, one of the reasons my lab started to work on this topic is because of the societal grand challenge that we have on antibiotic resistance. And so most of you may have sort of heard about what this problem is. So the problem is that we now have bacterial infection where the bacteria are no longer susceptible or can be eliminated by any antibiotics that we have. And this is partly because we've sort of used and misused and overused antibiotics over the last 20, 30 years, that the bacteria have become resistant to every known drug. And so as you can imagine, that this is a serious, serious problem. In some ways, this is the silent epidemic, right? Because again, when the COVID pandemic happened, it was, it happened over a 2-year period, and then we're sort of almost back to normal now. But this is a slow burn pandemic that's been happening over the last 20 years. And here's sort of a couple of data slides that says that if we really don't treat this antimicrobial resistance problem or drug resistance problem, the number of deaths from this would surpass cancer in the next 20, 30 years. Now, I'm going to sort of give you a couple of more examples to illustrate the reason why we're working on this problem. Here is a plot that shows you all the major antibiotics that have been introduced in the United States over the last, I'd say, 50, 60, 70 years. And this time point is when the antibiotic was introduced in the clinic, and this time point is when we see the first drug-resistant bacterium emerge in the clinic. And you'll see that typically through the 40s, 50s, and the 60s, it's a 10-year lifespan for a drug molecule, right? But as you get closer to the 2000s, as soon as you have a new drug introduced in the clinic, there is already spontaneous resistance present in the bacteria, right? So this is a serious issue. And the other big problem is if you look at the number of antibiotic molecules coming through the FDA pipeline, this is a dwindling pipeline, right? So this is the pharmaceutical companies are not investing in this. And so the pipeline is kind of running dry, and so it's a double whammy, right? On the one hand, we don't have enough new molecules that we're discovering, and the molecules that we are discovering don't really last too long in the clinic, right? And so that's the nature of the problem. And so what we decided to sort of do was, can we use these bacteriophages as potentially agents to kill multi-drug resistant bacteria? And so that was the vision that we're trying to advance in my lab. And now this idea is far from new. It's an idea that's been around for 100 years, and people have been using these bacteriophages, particularly from sort of former Soviet republics, Georgia and Poland. I mean, Poland is not part of the Soviet Union, but Poland too, where people had been using bacteriophages for well over 50, 60 years. In the West, and so here's sort of a picture of a lab in Paris where they had these bacteriophages. So this is from the 30s. This is an ad where they had used bacteriophages to treat all kinds of infections, and it was a marketed product about 50, 60 years back. Now this company, Le Laboratoire du Bacteriophage, eventually became L'Oreal, and so if you use this product, the origins of this company is making bacteriophage products. Okay, now what happened in the West was we discovered penicillin, right, around 30s and 40s. And the penicillin turned out to be a miracle drug, and we never really looked back, again, for the past 50 or 60 years, until, I'd say, the last 10, 20 years now, because of the antibiotic resistance problem. And so this is really what got us interested in the question. So I'm going to pause here for a second. I've gotten through a few slides, but I thought I might just pause for any general questions anyone might have.
Yeah, L'Oreal Paris, Food, Drug, and Cosmetic Act, indeed.
Yeah. Now they don't make bacteriophages anymore, but it started out as a bacteriophage company. Okay, so there are a couple of reasons why we pursue this. One is it's a precision drug, which means that each bacteriophage can target a specific pathogen and leave the other beneficial bacteria in us intact. They don't touch it. And so, for instance, if you, in our lower intestine, we have trillions and trillions of bacteria, which are actually fairly beneficial to us, and some of them are pathogens, and so these are like precision guided, where they can specifically take them out. They're evolvable, which means that if you have a bacteria that eventually becomes resistant to a bacteriophage, you can re-evolve the bacteriophage to become potent against that pathogen. And this is a cool property where it's auto-dosing, which means, again, you don't have to overdose the drug. When it finds its target, it kind of multiplies at sight. And so that's kind of a cool property that these bacteriophages have. And they're extremely abundant. I've already given you the number, 10 to the 31 phages on Earth, and so we'll never run out of them. And I'll sort of mention one story that really got us inspired into thinking about this. So this is a case of a couple. This is Tom Patterson. He's a professor at UC San Diego, and his wife, Stephanie Strathley, who also happens to be a microbiologist. And so they were vacationing in Egypt. So this story is about 4 or 5 years back. They were vacationing in Egypt, and he picked up some bacterial infection, and all the standard treatments failed, and so his health started deteriorating, and so they had to fly him back to Germany, and then nothing worked, and then they had to fly him back to California, and his wife, being a microbiologist, she looked up bacteriophages from that could potentially be targeted against the pathogen that he has, because he was sort of slipping out pretty fast. And then she managed to find bacteriophages from labs in Texas and a couple other places, and he was administered those phages as a last line of defense, and he actually made a miraculous recovery. And so in the last 4 or 5 years, I'd say, this story was inspirational to the field, and now people are thinking, how can we now push this beyond an anecdotal case and try and make this mainstream. And there are a couple of reasons why the phage therapy has not been adopted, and I won't get into the details of what these are. And one of the major issues is the activity of these phages in terms of killing pathogens is not nearly as strong as antibiotics. And so that's a major problem. And part of the goal of what we do in my own lab is to figure out how these natural phages work and how they interact with their pathogenic bacteria, and can we use that information to then design synthetic phages that are particularly effective against these multi-drug resistant pathogens. And one example which we've of success over the last couple of years from my own lab is, if you go to the CDC website, there is a list of the worst offenders in terms of pathogens. And there is this group which is called as, what do they call that? Highest threats or something like that. And so one of these bugs is resistant to 35 antibiotics. So it's really dangerous to even handle this bacteria. And so we've been able to design bacteriophages that can kill that bacteria. So we think there's some promise there, and we can potentially advance this as a general therapeutic against drug-resistant pathogens. So this is all the introduction about bacteriophages, and now I'm going to pivot to some of the space experiments. So I'll pause, but I'll take questions if anyone has any.
I'm just curious, if you find a bacteriophage that works, and you want to duplicate it to get a dose that will help somebody, how long does that take? How long does it take to grow a sufficiently large sample?
20 minutes.
20 minutes.
Yep.
Wow.
Yeah. If you have a culture in the lab, you can grow it out in 20 minutes, and so there is a lot of interest in that. So part of my lab actually works with the U.S. military and folks at Walter Reed, where they when soldiers are exposed to all kinds of pathogens in where they travel, they want to use this because you don't have to get it from some other place. You can just grow them right there on site and apply them. So that's one of the research efforts in my lab, is to work with the U.S. Army. Yeah.
Do you manipulate the phage, or he can hear me, I think. Oh, manipulate the phage by plasmid or interrupting, and there are RNA pages too, right? DNA, RNA, all of those. And so is it this kind of a machinery?
Yeah, so now we're getting to the weeds of how we do these things. So there are phages that use DNA as their genetic material. There are phages that use RNA as their genetic material. There's a single-stranded and double-stranded kinds. There's just many different kinds of these. And so the way we modify them is we synthesize DNA outside that has whatever changes we want to make, and then we sort of swap that into the phage DNA. And so the new phage progeny that gets made is different from the phage that's. Yes, we make modifications, correct. Okay.
But they use phages to engineer bacteria.
They do, and so you can design phages in such a way that it does not kill the host. So you can take out elements of the phage where it simply gets in and delivers DNA, and so we can do that too. Right.
And I just want to quick remind everyone that, yeah, the guests joining us online, listening to us via YouTube, the only way they can hear your questions is if I can relay it through this microphone. So, yeah, thank you very much. Go ahead.
Okay, so I'll pivot to the space stuff. So what this happened about 2.5 years back, just before the pandemic, and we were in touch with this agency, the Department of Defense, called the Defense Threat Reduction Agency. It doesn't matter what they do. They were working with NASA, and there is a strong push by NASA and some of the other agencies to start conducting experiments at the International Space Station. And so these could be simple sort of biology experiments, experiments in physics or chemistry, material science or whatnot. And so there's a push to, and so you might have heard of these national labs that we have here. So Argonne National Lab is in Chicago. There's like 7 or 8 of these in the United States. And the International Space Station actually happens to be a national lab. And so one can send samples up there and the astronauts will do experiments for you. And so we were in contact with the DoD agencies, and they said, well, there's an opportunity to run some biology experiments. What would you like to do? And I said, well, there's a microbiology 101 experiment that I'd like to do, which is simply to ask how do phages and bacteria interact in space. And so that was sort of the motivation. And we really wanted to sort of try and answer 2 questions. How do basic principles of physics and biology, which are presumably different because of changes in gravity versus microgravity, and how does it affect these interactions? And perhaps the longer term vision was, when we get to the point where we start sending people away for long-term space travel, a key component of how we need to, for quote unquote, sort of package them for long-term space travel is how the microbiome works. And so the microbiome is, again, the collection of microbes that lives in your lower intestine. And so they have a huge role in how your food gets digested, how your immune system works, how your inflammation in your gut area works. And so it's a sort of a pivotal component of overall human well-being. And the microbiome is a mixture of bacteria and phages. And so as we start thinking about sending people sometime down the road for long-term space travel to Mars, well, how do we know how phages and bacteria interact, and how might that shape what the microbiome looks like? So that was sort of the pie-in-the-sky vision for why we were doing this. Now, we're not close to addressing this question, so we're still taking baby steps. So that's what we've did in the last couple of years. Okay, so when we finally got approval to do this project where we would send samples up to the ISS, it turns out that every project gets a mission patch. I don't know if you guys have heard what a mission patch is. So every project gets a mission patch. So this is the kind of stuff, the astronauts, not the astronauts, I think people who are working on this research project, they put this on their shoulders. And so we were asked to design a mission patch. And one of my graduate students, she's exceptionally talented with art, and so she made this. And I'll just point out a couple of things. This is the bacteriophage, as you can tell. Here is the DNA. So this is the rocket, and you can see Bacteriophages in there. And so here's the bacteria in the lower part here, and this is the International Space Station. And so I thought this was a pretty cool image that she designed. And so this became our mission patch, and they printed this out, and then we were flaunting this around in the department for many months. Oh, they love this kind of stuff. Yeah, they ate this up. I'll point out a couple of key stakeholders and partners that for this project, and they're all, this out here, but I just sort of walk through them. This, of course, is University of Wisconsin-Madison, my employer. So this is NG-13. This is Northrop Grumman. So this is the company that actually made the rocket that sent our samples up to space. And this is what that rocket looks like, NG-13. So it's pretty cool. I'll show a couple other pictures a few slides down. This is the agency that supported our research. This is it's a Defense Threat Reduction Agency. This is part of the Department of Defense. Their key mission is to protect us against chemical and biological and radiation agents, but they also support other kinds of research. So this is the International Space Station, and as I said before, it actually turns out to be a national lab. So once you get a project, you can send samples up, and there are people who will perform the experiments for you. This is the company that helped us sort of make the samples, because it turns out that you can't just simply put stuff in a box and send it up. There's a lot of quality control that needs to happen before you can ship samples up to space. Everything has to be triple wrapped or quadruple wrapped, and you have to do temperature tests, pressure tests, all kinds of things. And that's just a simple vial of like bacteria. It goes through a lot of quality controls before they would send it up. And so these guys helped us sort of meet those conditions. And this is, of course, my own lab. Okay, so again, I said, we were trying to get at some basic principles of physics and biology through these experiments, and it was, there are two things that are fundamentally different between, terrestrial conditions and ISS. One is radiation, but since it's inside the ISS capsule, there isn't a whole lot of radiation. So the major difference then is microgravity, right? And you've seen the animation, how the bacteriophage interacts with the host. And so how the phages migrate through solution and find their host is really the fundamental thing that drives how well they can infect their host. And once gravity changes, right, so the mixing properties change, how the cell sediment changes, how they diffuse through solution changes, and the pressure they might experience changes, right? So these are some of the variables that are different between ISS and Earth. And so again, these are the experimental parameters that we were hoping to test. I forgot Phil's name. Okay, well, Phil is here. Phil and Shrutikar are the two graduate students in my lab who, again, did all these experiments. And so you can see the rocket back there. So this is a launch station, which is 3 hours east of DC. I forget the name of this place now.
Wallops Island?
What was that?
Wallops?
Yeah, Wallops Island, correct, yes. So there are two places where you can launch rockets. One is Wallops Island, the other is Kennedy down in Florida. And ours was from Wallops. And so Olivia and Heath are from Rhodium Scientific, and this is RP also from Rhodium Scientific. So we were there for the rocket launch. And it turns out, again, I had not seen one of these before. They would scrub the rocket launch like 10 seconds before launch. And so we spent a lot of money to fly all for the grand finale. And so we were there. So this is about a mile and a half away from the rocket. And we were like, it's going to take off at like 3 o'clock in the afternoon. And then it was 30 seconds, 20 seconds, 10 seconds. And then nothing happens. And then we were like, oh my God, we spent like $5K for the rocket launch. And then they scrubbed the launch. And then they tried it the next day, they scrubbed it again, and I was like, well, I don't think I'm going to wait for this any longer. I'll go back and watch this on YouTube. And so I had to fly back to Madison, and then they eventually sent the rocket up 4 days after. So this is the picture of the Northrop Grumman rocket. And so it's also surreal that the box of samples that was lying around in my lab like a couple of days back is now up on that rocket. So it was quite a moment for us. Okay. So this is the astronaut Andrew Morgan. Apparently, we're told that this is when he's taking our sample out, but I don't know. We'll just have to take their word for it. And the way you design these experiments is you can make them. so again, these are all things that I learned as we were doing these experiments. You can make them do complicated things. So if it's if the procedure is like, you know, take it out and mix this with that and shake this, they won't do that. Or it's the clearance required to make that happen is at a whole nother level. So what they would do is they would take the box from the freezer, leave it outside for like 15, 20 minutes, and there's something that shakes, and they'll put it back. So that's all the astronaut would do. Take it out, leave it on something, and then put it back in 20 minutes. And if we added a step where it said, you know, the astronaut has to sort of tilt it around a few times, they'll just reject it right there, right? So it was, I mean, you know, again, there are 5 people doing experiments for like 50 projects, so, you know, maybe it should be expected that that's all they'll do. Okay, I'll stop here at this point, and I'm going to get to a few data slides, but I don't know how much time we have. Any questions so far?
We have plenty of time, by the way.
Okay. Any questions? All right. Okay, so the way these experiments, so I said earlier it's a microbiology 101 experiment. So it's a really simple experiment, right? Because we're asking if we mix the bacteria with the bacteriophage, how is it different in space? And again, it turns out that you've got to run these experiments with appropriate controls. And what that means is that you've got to have conditions where you replicate the exact experiment, but on Earth conditions, and then compare the results with the space experiment. And this was a fairly onerous process. Again, this is our sample box. So we were given a box which is about this size and this deep, and you could slot in like 25, 20, 30 samples, and that's it. That's all we were given. And so the way we went about this is we would prepare 2 samples under exactly identical conditions. We would split them. And one would be the space sample, the other would be the terrestrial control sample. And both the samples would be frozen because the space sample would be, you know, has to go through a cold chain all the way to the ISS. And we would freeze the terrestrial samples to maintain the conditions to be as close to the space condition. And then once it's frozen, it goes up to ISS. And then there it stays incubated again in the freezer for some period of time, and when the astronaut decides, he or she takes it out, leaves it outside for 37 degrees, and again we replicate these exact same conditions on Earth. And like I said, you know, we were measuring a few different variables: sedimentation, diffusion, hydrostatic pressure, and in general how phages and bacteria interact under these conditions. And once this experimental period is done, it was about 25 days. And you know, again, with some frequency, they send these rockets up to stock the ISS and then bring back, I don't know, trash or whatnot. And you know, our samples come down with the trash, and then they ship the samples back to us. And so we're the box in which the samples went up and came down. That's like a that's right up at the topmost level in my lab where people can, we're very impressed to hold that. It's a shrine almost in my lab to see that box, and it's just it's a totally insignificant box. And so then we do the sample processing and analysis, again under identical conditions. So now I'll get into a little bit of technical detail where we talk about what the experiments are. We do sort of 3 kinds of experiments with this, again. So again, terrestrial conditions and space samples. We first simply count the number of phages that are present in the sample at a couple of different time points. Next, we sequence the DNA of the phages, and we ask, are there any mutations in the DNA, right, which is different. And this third experiment is a little more complicated, where we make large mutations to the phage and ask how they're different. And if we have time, I'll try and get to this, because this is actually the coolest experiment. Okay, so what's the first experiment? We simply count the number of phages and bacteria. I apologize for the bar plots, but you know, at some point we've got to get our hands dirty and look at data. And so this is these are 4 time points that we measured, right? We're asking how many phage particles or phages are present after 1 hour, 2 hours, 4 hours, and 23 days. Again, We picked these time points only because we were only given a limited box size. And you'll see that sort of the phage count is relatively steady for the short period, and then it drops after 23 days. This is the terrestrial conditions, right, gravity. Now, if you compare what happens to microgravity, again, the 1 hour and the 2 hour and the 4 hour time point is more or less sort of steady, but the long term, the 23-day time point, there is a pretty steep drop. So we lose a lot of phages going from 4 hours to 23 days. So just sort of keep that in mind. Now, the other important thing is, so we've looked at this plot where we've only looked at what the phage count looks like, right? So there is no bacteria present. This is simply the phages. How long do they live if we keep them for 23 days? How many of them live if we keep them for 23 days? Now, this is the second experiment where we mix the phage and bacteria. And as you might imagine, once it finds its bacterial host, it's going to multiply and make many, many baby phages, right? And in this case, you'll see that, at 1 hour, again, this is gravity, so this is Earth conditions. 1 hour, 2 hour, it's about the same, and boom, there is this explosion because it's making many, many phage progeny, and then it goes up. And after 23 days, it kind of dies off because, they generally degrade over time. Now, if you look at what this exact same experiment, if you compare that with the space sample, the microgravity sample, you'll see that it's steady for 1 hour, 2 hours, and 4 hours, but at 23 days there's a steep rise, right? And so one conclusion that we could draw from this, I'll toggle back, for the Earth sample, the replication, which means it's made productive interactions with this host, has happened within 4 hours. But for the space sample, the productive interaction has taken a much, much longer time frame. And so one sort of key takeaway for us based on this simple experiment is, phages diffuse more slowly under microgravity conditions than Earth. Now, you might argue that this is, what would you would have expected this even before you did the experiment, but it turns out that no one's actually done this experiment. So this is the first demonstration that it is indeed the case that they diffuse more slowly or there's lesser tumbling. So this is one piece of data. And again, this is a very simple microbiology experiment, but to do this systematically with ISS and Earth is a step forward. Now, the second experiment we did was we sequenced the genome. Again, the phages have their DNA, and these are highly adaptable, which means that they can pick up mutations that facilitate their survival. And if they have, over a period of time, if you see a mutation that stays so a mutation is simply a change to its DNA sequence, right? And if it makes mutations to its DNA, these are often beneficial mutations that the phage is making, right? So if whatever helps survive those conditions, it'll make those mutations. And so what we did was we would take the natural phage that we have under Earth conditions, we sequence its DNA, and then we take the phage that's gone through the whole space experiment and we sequence its DNA, and if we ask, are there any differences in the DNA sequence? And so here's a cartoon version of the phage. There are about 40 different genes. Again, doesn't matter what they are, but this is the entire DNA of the phage. And so what you'll see, these red and blue spikes are bars where there's more mutations in the space phage than the terrestrial phage. And some of these genes are what are called a surface protein. So again, you can imagine that this is the bacteriophage is like a shell with proteins on the outside, and these proteins on the outside are what help the phage stick to different things. And again, we see a larger preponderance of mutations in the surface proteins, and one takeaway from that is that these surface proteins, mutations to these surface proteins perhaps benefit the phage because as it tumbles through a little slowly in ISS, maybe this mutation is like a Velcro. It kind of helps it sticks to it, to its bacterial host a little better. So any questions on the data so far? Okay. So I'll conclude by talking about the third experiment. So this is the most complicated experiment we've done. And so we're going to have to delve just a little deeper into how these phages work to understand what this experiment is. So I showed you this picture. This is how the bacteriophage interacts with its bacterial host. And if you sort of zoom in a little closer, right, this is the bacteria surface, right? This is this part over here. And there are these proteins on the surface called receptors, right? And again, you might have heard of this term through the COVID-19 because as COVID evolved, it picked up mutations that allowed it to interact with different human host receptors. It's the exact same idea with bacterial viruses too. And so this bacteriophage finds its receptor, and that's how it sort of gets into the host. So this is what that looks like. This is what the part that is the feet of the bacteriophage. This is the shape of it in atomic level detail. Again, the details don't really matter here. Now, what is important is that this whole thing here is essentially a sequence of amino acids. It's just it's like a polymer, right? It's a polymer of amino acids. And what we do to this amino acid sequence is that we introduce many mutations. And so these are thousands and thousands of mutations that we introduce into the feed. So I won't go through the details, so I'm just going to step this. And so the idea here is that you make a population of bacteriophages where each bacteriophage has a unique change to its DNA sequence. And the idea there is that we're almost sort of supercharging evolution, right? We're making, the way evolution works is it makes mutations, and whatever mutations are beneficial, those stay, and the mutations that are not beneficial kind of fall off. And so in the lab, we can simulate this experiment where we can introduce many mutations into the bacteriophage and ask which mutations were beneficial and which mutations were not beneficial to the bacteriophage. And so you might imagine, so let's say you have 3 different mutations. The yellow mutation was not beneficial to the bacteriophage. And so when you throw that on the bacteria, this phage will kind of die because it's not making a productive infection. If you have a bacteriophage with a mutation that is, it's about the same fitness level as the native bacteriophage, then its numbers won't really change. But if you're making a bacteriophage with a mutation that is beneficial, right, it's able to stick to its host better, it's able to do something better, then it's going to make more baby phages, right? So it's really a simple evolution experiment. And so we do this large-scale evolution experiment, and no one's done this at this scale in the International Space Station, so that really sets this experiment apart. And I'm just going to quickly jump to the end of this presentation. So this is a complicated heat map to look at, and I'm not going to try and walk you through this, but this is the polymer sequence. Each alphabet is a certain type of chemical in that polymer, and here are the changes that we make, right? So the takeaway here is that the red blocks are mutations to the bacteriophage that are beneficial. The white blocks are mutations to the bacteriophage that hurt the bacteriophage, right? And so the one thing you might see is that if you compare the microgravity to the gravity, they're not identical patterns. So even if you don't get any of the details, the one thing you can take away is that this barcode, if you will, looks different from this barcode, right? And so what this told us is that there's something about the bacterial host that's changing under microgravity, right? Because the bacteria is not accustomed to microgravity either, right? It experiences normal pressure under Earth conditions. And so when you take it to microgravity, it's kind of stressed out. And so when it's stressed out, it's changing the proteins that it's showing on its surface. And because it's changing the proteins it's showing on its surface, different mutations to the bacteriophage help the bacteriophage get inside. Does the logic here make sense to people? Right? So as you stress the bacteria as it goes up to microgravity, its surface profile is changing. It's making different receptors now. And when it's making different receptors, different mutations in the bacteriophage let it get inside. And so that's why this pattern is different from this pattern. And so that was sort of the conclusion that we had was that the phages are sticking to different surface receptors because the bacteria is stressed and is fundamentally different from what it would be under Earth conditions. So this is as far as we've gotten with this experiment so far, and I'm just going to conclude by sort of quickly saying that this is the simplest microbiology experiment. This is the kind of stuff that a high school student would do, but it's funny that no one's done this in space conditions. So this is our first step in that direction. The second point is our goal is to push the complexity of this experiment, where we go from one type of bacteriophage to one type of bacteria to many types of bacteriophages and many types of bacteria, which more closely mimics what we have in our human gut, right? And so the complexity of this thing, this experiment has to go up. And again, as I said initially, the goal is, can we then design these phages that will allow us to, that'll permit long-term space travel. So with that said, again, this is my research group. These are the people that have given us money over the years to support the research that we've done. Again, these are the folks that do all the cool stuff. I just get to give the talks here. So with that, I'll stop. Thank you. Yeah.
Just curious, do you have any more space missions in sight? And number two, this is totally different. The bacteriophages and the bacteria kind of work together.
Yep.
Which came first? Did the viruses come first that created the bacteriophages, or what was the evolutionary sequence to end up with the bacteriophages and the bacteria?
Yeah, well, the second question is very hard to answer, but one can speculate. So we don't have any space experiments lined up just yet because we got to publish this. This is all unpublished, so we're writing this up and we'll publish this in the next couple of months. Once we publish this, then we'll dangle this in front of the granting agencies. We'll get more money and spend it, and that's the way this vicious cycle works. So yes, we will do space experiments, but we don't have anything lined up. So the second question, what came first? The phages certainly came first, right? Because it's only genetic material. The bacteria is a much more complex system. They have metabolites, chemicals, enzymes. It's a vastly more complex system than bacteriophages are. Now, then how did that evolution actually happen? That's a super hard question to answer.
Can you go back to the, you call them the barcodes with the white and the red?
Yeah.
I just have a question about that. And I don't know how well I'll be able to articulate it.
Oh, that's okay. I wasn't sure if I should. I put a lot of thought into whether if I should show this thing or not.
Oh no, it's really good.
This is a complicated experiment.
So the white are essentially mutations that were less fit.
Correct.
And red are more fit.
Correct.
Intuitively, I would think that there would be more red dots in microgravity than gravity, because in the control experiment, the phages are already well adapted to their environment, to their host, and anything that changes that would theoretically add more opportunities to improve.
So let me answer that question this way.
Does that make sense?
I see where you're going with this, but let me answer it this way. Under gravity conditions, let's say there are 4 types of receptors that the phage uses to get inside. It's just a lock and key mechanism, right? Now, when the bacteria gets stressed out, it might stop expressing 3 of those and might only have 1 of those, right? Because it's turning on other things to counteract the microgravity stress. And so the mutations, there are fewer ways the bacteriophage can now get inside. That the key is going to fit the lock in fewer ways. So that's our speculation, that the surface profile of the bacteria has changed because it's stressed out. That's the way we're rationalizing this.
Now, would the next step be to test that by creating some sort of experiment that tests the bacteria itself without the virus and measuring its receptors when it comes back?
Yeah, so that'll be the obvious follow-up thing to try out. The problem is
you can put me as a co-author.
yeah, I will, thank you. But I'll tell you why we can't do the experiment. So the problem is, to do that, we got to send samples up to space again, right?
Yeah.
So the microgravity is stressed. So it turns out radiation, you can simulate that condition in the lab, right? You can create, there are radiation sources that mimic space radiation. But microgravity is, people have these spinning systems that mimic microgravity, but it's not really quite the same thing. So how do we stress the bacteria with microgravity? That's a non-trivial problem.
One experiment that I thought of, because I was thinking the same question, you could study the rates of fermentation of alcohols in microgravity and do a little micro brewing up in the space station and see how it works out.
I don't know how I would swing that with the funding agencies. You can get New Glarus to pay for that. Any other questions? I mean, it doesn't. Sorry, I'll send this.
Yeah, that's kind of the funny thing is, yeah, how does the bacteria work in that? And one thing when I was thinking about Dan's question, it occurred to me, it'd be interesting to see, if the rate of mutation in the microgravity, kind of like with the other data where there was kind of a delayed reaction where in microgravity, once it got going. So maybe the rate of mutation might take.
So we are sequencing the bacteria too. I don't have the data yet, but we're going to see if it's different or not. Again, the problem with microgravity is that if we want to go back and test something, right? This is like a one-way street, right? So there's no follow-up, but. I mean, it is what it is.
Yeah, it takes years and then funding and the whole process.
Correct.
Oh, yes, you're gonna.
I was just curious, you had 1 to 4 hours and then 23 days.
Yeah.
And especially, that's a long pause.
Yep.
And so I'm curious, for the microgravity where it was lower and then suddenly jumped up, do you have any sense at all what's going on in that roughly 22.5-day period?
Yeah.
I mean, could it be that it spikes and then goes back down?
Oh, absolutely.
I mean, this is 2 points make a wonderful straight line. But
No, you're totally right. I mean, so the part where I said it's 4 hours and 23, there could be a lot of crazy things going on. There could be cyclical for all we know, right? It's again only because of space constraints we had to pick a time point, right? So we said, under normal conditions, these the life cycle is about 30 minutes. We said, okay, we'll let about 3 or 4 life cycles happen, and then we'll pick something which is 20 days out and see if it's different. But you're right. I mean, it's more granular measurements would be. We were only given a box, unfortunately.
23 days is kind of an arbitrary number.
It was we had to pick that number because the spacecraft that was bringing the trash back was scheduled for a certain time, and so we're like, okay, we'll go with that time point. Yeah,
You're in the recycling.
that's right, exactly. Yeah.
How do you stop at time point? Do you freeze them?
Yep, they take it and they jam it back into the -90 freezer.
And then that's the 4-hour time point, because after 4 hours it was dropped back in the freezer.
Yep, correct.
Back to you is what you're analyzing. What samples come back having gone through SACE or have stayed here all the time at these periods? You do the sequence of that combined product. So the phage would have had to find the bacteria. Are they separated and then you combine them and then this goes on?
No, so what we do is they're pre-mixed, but they're pre-mixed and instantly frozen to -80, and then we send them back, send them up. And so the way we do the control experiments is they give us the exact, so we ask them to take it out and put it back in an hour. Sometimes it's an hour and 20 minutes because we can't control the astronaut's time. But they will record that. They'll tell us what they did. Yeah, they'll keep track of that. They'll tell us what temperature they incubated. And so we'll get actually fairly high resolution data on how they performed that experiment, and then we'll try and mimic that down here. Sorry.
Sorry. I was just wondering, you were talking about mimicking what they did in space. So when you're doing the control part, you're doing that after they perform
Correct.
the experiment in space,
Correct.
so that you can be sure to
Exactly.
have the same conditions. Okay.
Yeah, that's right. I mean, it's as close to a mimic as we can do, right?
All right, are there any additional questions?
I'll maybe mention one because I want to circle back to the antibiotic stuff. One of the fun things that actually came out of this experiment was the mutations that were highly enriched in the microgravity. I forget which one it is. My student will know. These mutations in the phages could actually kill one of the lettuce E. coli pathogens. So, which was sort of an interesting. It was not what we were expecting. It was somehow enriching for mutations that the native phage could not kill that bacteria, but the mutants that came from microgravity, they were actually good at killing the foodborne lettuce E. coli. And we're like, whoa, there we go. There's that is serendipitous. That's right, exactly.
I don't know about the food packs that they send up to the space station, but there might be. Well, Vatsan, thank you so much. This is a very fascinating topic and a very
Thank you.
Unique experiment. I think this is one of the most, as far as microorganisms like viruses and bacteria goes, I think it's really fascinating to be able to learn about how they interact and behave in microgravity and outer space. So thank you very much on behalf of Madison Astronomical Society. This is a token of our gratitude for such a great and fascinating presentation, and
Thank
we
you
really
very much,
enjoyed
Lawrence.
having you here.
Thank you. Thank you all for coming. Thank you.
And before we go, I'd just like to tell everybody a little bit about next month's presentation. Our next meeting is going to be on Friday, March 10th. And as we discussed earlier, we would like to have a General Assembly vote on the proposal that Jürgen presented earlier. But we're also going to have a presentation by the director of UW Spaceplace, Dr. Jim Lattis. And the title of his presentation is Washburn's Other Observatory, the Student Observatory, which was active from 1879 until 1960. So I guess he's done some research on the origins and history of the UW Student Observatory, and he'll give a presentation on it next month on Friday, March 10th, right here. Well, thank you so much, everyone, for coming, and we'll see you next month.